Do you hear me? Do you able to hear me or not? Okay, I have taken your presentee, but do you able to hear me? Yes or no? Okay. Okay, okay. So what we have seen yesterday? What we have learned yesterday? Okay, what we have learned yesterday about the facilitated diffusion and passive transport. Passive transport is also known as transport is also known as what? This is my question. Passive transport is also known as answer as early as possible. Passive transport is also known as what? Passive transport is also known as simple diffusion. That is the transport. Yes, Jayashree have answered it. Very good, Jayashree. Only Jayashree have able to understand yesterday, I think. What about others? They are only mentioning their presenting. Present. Simple diffusion is also known as passive transport, that is the transport of molecule from higher concentration to lower concentration, that is molecule transfer only from higher to lower. There is no ATP molecule is required as a source of energy, and there is no requirement of carrier molecule or protein molecule for the transport of substances. Only molecule transfer from higher concentration to lower concentration up to the concentration becomes equal on both sides of the membrane. This is the simple diffusion, which is also known as the passive diffusion. Then we have seen facilitated diffusion. Facilitated means diffusion is facilitated. Facilitated diffusion means diffusion is carried out with the help of presence of some proteins. The proteins are present in the phospholipid bilayer of the membrane. That protein passes the molecule from outside of the membrane to inside of the membrane. That is, facilitated diffusion does not require any ATP as an energy source, but it require facilitated diffusion require the presence of membrane protein. That membrane protein allows the passage. That membrane protein carry out the binds to the molecule to be transported and then passes it into the membrane. Okay, then it shows stereospecificity. What is meant by stereospecificity? Are you hearing me? Are you hearing me? Facilitated diffusion require, okay, Facilitated diffusion require presence of protein, so that protein gets saturated with the substance to be transported. एका स्पेसिफिक लेवल पर्यंत substance pass होईल, त्याच्यानंतर there will be no transfer of molecule because it gets saturated. So it shows stereospecificity. What is meant by stereospecificity? That is only two of the one isomer, D or A isomer, is able to transfer by facilitated diffusion. So there are a number of characters different from the facilitated diffusion. Yesterday I told you, write down the homework in between the difference between the two, that is facilitated diffusion and passive diffusion. How many of you have written that test or written that homework? How many of you have written? Okay. So today I am going to start the today you will be able to see the PPT because PPT is now appearing on YouTube. So today you will be able to see that yesterday's PPT. So you write down from that PPT. So today we are going to start with the primary active transport. So primary active transport. This is next to the transport mechanism, that is after passive diffusion, after facilitated diffusion, next is the primary active transport. So what is primary active transport? Now pay your attention properly because this is little bit hard topic as compared to the previous one. That's why I am taking this second unit first, because this unit second and third, these are the hard as compared to ACC. That's why I am conducting this. Primary active transport is the transport of substances through the membrane against the concentration gradient. What is it? It is the transport of substances through the membrane against concentration gradient. So it depends upon supply of metabolic energy. Depends upon the supply of metabolic energy as a ATP. ATP acts as a energy source for the transport of substances against the concentration gradient. Active transport requires the specialized protein carrier to bind with the substance and transport it. So what is primary active transport? Primary active transport requires the specialized protein carriers. So here again protein carriers are required to bind with the substance and transport it across the membrane. Again it shows saturation kinetics. Saturation kinetics म्हणजे काय? Membrane protein get saturated, presence of carrier protein in the membrane, they get saturated with the substance to be transported, and then it shows saturation kinetics, that is again increase of the substance to be transported. There is no increase in the transport rate from outside to inside. Ion transport is the most important type of active transport. Ion transport takes place by the active transport mechanism. So active transport mechanism requires, it takes place against the concentration gradient. It depends upon the supply of metabolic energy as ATP. It requires the specialized protein carrier to bind with the substance and transport it. Then it shows saturation kinetics. Saturation kinetics means carrier protein gets saturated with the substance. Most important example of the active transport is the ion transport, that is transport of ion with the help of P-type, V-type and F-type ATPase. These are the primary active transporters which carry ion from outside to inside of the membrane. समजा बाहेर तुमचं सोडियम क्लोराइड आहे, तुमच्या न्यूट्रन मीडियामध्ये, your न्यूट्रन मीडिया contains sodium chloride, then media have specific pH, that is hydrogen ion concentration, then medium have other type of cations, anions. So that ion transport takes place with the help of this PVF type ATPase. These are the examples of primary active transport. ATPase are the membrane-bound ion channels that couple ion movement through the membrane with the synthesis or hydrolysis of nucleotide ATP. What are ATPase? ATPase are the membrane-bound ion channels. These are the membrane-bound ion channels that couple the ion movement through the membrane with the synthesis or hydrolysis of nucleotide ATP. म्हणजे ATP hydrolysis करून आयनचं मूवमेंट करतो कोण ATPases and that ATPases are the membrane bound ion channels. These are the ion channels or ion proteins present in the membrane. So there are four main classes of ATP powered pumps based on the functional differences. They are P class, V class, F class pumps and ABC superfamily transporters. कोणते कोणते P class, V class, F class pumps and ABC superfamily transporters. So ATPases are membrane bound ion channels. These are the membrane bound ion channels that couple the ion movement through the membrane with the synthesis or hydrolysis of ATP molecule. There are four main classes. These are the P, V, F and ABC superfamily transporters. So first is the P-type ATPase. The family of ATP driven cation transporters. P-type ATPase काय आहे? ATPase, these are the cation transporters. ATP ड्रायव्हन म्हणजे ATP युटिलाइज करून P टाइप ATPase transfer the cations across the membrane. They are reversibly phosphorylated by ATP, hence they have the name P type. Phosphorylated, phosphorylation होतं त्यांचं म्हणून त्यांचं नाव काय आहे? P type ATPase. P type ATPase are the P stands for phosphorylated. Phosphorylation factor किंवा P type and ATPase are the ATP requiring transporters. Transporters means ion channels which require ATP. Hence these are known as the ATPase and P means phosphorylated. That is the ATP get phosphorylated, hence the name is P type. Phosphorylation forces the conformational change that moves the cations across the membrane. Phosphorylation forces conformational change in that transporter that moves the cation across the membrane. त्याच्यामुळे काय होतं? कॅटायन मेम्ब्रेन मध्ये पास होतात. फॉस्फोरिलेशन होतं कशाचं? ATPचं. ATP get phosphorylated which causes conformational change in that transporters and that moves the cation across the membrane. So all P class ion pumps, they possess two identical catalytic alpha subunit each containing ATP binding site. काय आहे? What is the structure of P class ATPase? P class ion pump possess two identical catalytic alpha subunit. त्यांच्यामध्ये they are structurally made up of two identical catalytic alpha subunit each containing ATP binding site. Most of them also have two smaller beta subunits. त्यांच्यामध्ये काहींमध्ये beta subunit पण असतं which usually have regulatory function. They are widespread present in eukaryotes, in bacterial and bacterial plasma membranes. कोणामध्ये असतं? जास्त करून eukaryotic and some bacterial plasma membranes. They transport a variety of compounds including ions and phospholipids across the membrane. काय करतात ते? They are responsible. They are transporting variety of compounds like ions and phospholipids across the membrane. Example: sodium, potassium, ATPase in the plasma membrane. Sodium, potassium, ATPase, that is sodium plus K plus. So these cations are transported by the sodium potassium ATPase through this plasma membrane, through the plasma membrane into the cell. So this is the P type ATPase. Okay. Family of ATP driven cation transporters. These are the P type ATPase. They are reversibly phosphorylated by ATP. Hence phosphorylated, hence name is a P type ATPase. Phosphorylation forces the conformational change Because of the conformational change, cations passes across the membrane. All the P-type ATPase, they possess two identical catalytic alpha subunit containing ATP binding site as well as the two smaller beta subunit which have the regulatory function. They are present in eukaryotes and in bacterial plasma membranes. They transport a variety of compounds including ions and phospholipids across the membrane. Sodium potassium ATPase is the example of P-type ATPase. Okay, do you have any difficulty in all these? Okay or not okay? Do you have difficulty in P-type ATPase? P-type ATPase are good, no difficulty. So you are paying proper attention. Okay, next is the V-type ATPase. V for vacuolar H plus ATPase, that is the V for vacuolar H plus vacuolar type ATPase. They are the class of only H plus ion transporting ATPase. V type काय करतात, फक्त H plus ला ट्रान्सफर करतात. P टाइप काय करतात, जनरली सोडियम पोटॅशियम आणि कॅटायन्स जे आहे त्याला ट्रान्सफर करतात. व्ही टाइप्स आर ओनली फॉर H plus ion transporting ATPase. They couple the energy of ATP hydrolysis to transport protons. ते काय करतात, ATP हायड्रोलिसिस करून एनर्जी जी इव्हॉल्व होते, that energy is utilized for the transport of proton, that is H plus ions across the membrane. These proton pumps are present in the membranes of lysosomes, endosomes and in plant vacuoles. Here this H-type H plus vacuolar V-type ATPase are present. They are present in the membrane of lysosomes, endosomes and in plant vacuoles. They function to acidify the lumen of these organelles. ते काय करतात, तिथे acidification H plus ion येतो म्हणजे काय होतं, increase in hydrogen ion concentration means results into the increase in acidity of the lumens of these lysosomes, endosomes and plant vacuoles. A low lysosomal pH is necessary for optimal function of the many proteases, nucleases and other hydrolytic enzymes in the lumen of these organelles. या ऑर्गॅनेल्स मध्ये लो पीएच पाहिजे, कशासाठी, फॉर द ऍक्शन ऑफ मेनी एन्झाइम्स लाईक प्रोटीएजेस, न्यूक्लिएजेस or other hydrolytic enzymes which are present in the lumen of this organelle. So V-type ATPase is also known as vacuolar H plus ATPase. In order to become acidic, movement of proton must be accompanied by movement of equal number of anions, Cl minus chloride ions in the same direction. Movement of equal number of anions. Anions म्हणजे H plus बरोबर Cl minus ion पण मध्ये जातात, सेम डिरेक्शननी मध्ये जातात. Movement of equal number of different cations in the opposite direction. पण मग H plus जर मध्ये जाईल, Cl minus पण जर मध्ये जाईल, तर काही कॅटायन जे आहे ते मात्र बाहेर येतील. So these V-type ATPase are involved in vital intra and intracellular processes. काय करतं की vital intra and intracellular processes are carried out by the V-type ATPase. I will now repeat again. V for vacuolar H plus ATPase. Vacuolar means these are present in the vacuoles. जे पण सेल व्हॅक्युअल्स आहे लाईक lysosomes, endosomes, plant vacuoles, where these V-type ATPase are present. They are the class of only H plus ion transporting H ATPase. H plus ions त्याच्यामुळे ट्रान्सफर होतात. They couple the energy of ATP hydrolysis during the transport of proton. Proton transfer करताना ते ATP energy hydrolysis करून ATP energy वापरतात. These proton pumps are present in the membrane of lysosome, endosome and plant vacuoles. They function to acidify the lumens of these organelles. म्हणजे त्यांचं मधलं जे लेअर असतं त्याचं ते ऍसिडिफिकेशन करतात. कशासाठी ऍसिडिफिकेशन करतात? बिकॉज द proteases, nucleases and other hydrolytic enzyme present in that organelles. These enzymes require the acidic pH for their optimum activity, and this optimum activity in presence of acidic pH is performed by these organs. So these organelles have V-type ATPase. So in order to become acidic, movement of proton it is accompanying by movement of anions, Cl minus ions in the same direction, or movement of equal number of different cations in the opposite direction. म्हणजे एक्सचेंज होतं. कॅटायन्स आतून बाहेर येतात, हे जे आहे H प्लस बाहेरून आत जातात. त्याच्याबरोबर सायमल्टेनियसली Cl मायनस पण जातात. आता असं नाही आहे की हे व्ही टाईप एटीपीएस सेलच्या बाहेरच्या मेम्ब्रेनलाच प्रेझेंट असते, असं पण काही नाही. एखाद्या वेळेला सेलमध्ये पण आता जर सपोज व्हॅक्युओल्स हे जर मध्ये असली, एंडोझोम्स हे सेलच्या मध्ये असतं सायटोप्लाझम मध्ये. If it contains in cytoplasm, then from the cytoplasmic organelle, cytoplasmic content मध्ये जे H प्लस आणि सेल मायनस असेल ते तिथल्या तिथे सेलच्या मध्ये जाईल. सेलमध्येच मधल्या स्ट्रक्चर मध्येच ते ऑर्गन्स असतील एम्बेडेड असतील तर सायटोप्लाझम मधून ते त्या ऑर्गन्स मध्ये जाईल आणि सायमल्टेनियसली मधून बाहेर अपोजिट डिरेक्शनला कॅटायन्स येतील. म्हणजे इट इज नॉट नेसेसरी एव्हरी टाइम दॅट दिस आर प्रेझेंट इन द आउटर सेल मेम्ब्रेन. All these proteins, all these carriers, every time it is not necessary that they should be present at the outer surface of the cell. आउटर सरफेस वरती मोस्टली कोणते प्रेझेंट पाहिजे की जे बाहेरचे मॉलिक्यूल सेल मध्ये पास करतायत ते. एक्झाम्पल फॅसिलिटेटेड डिफ्युजनचे किंवा पॅसिव्ह ट्रान्सपोर्टचे जे आहेत ते. पण सेल मधल्या मध्ये जे ऍक्टिव्हिटी चालतात तिथे पण ट्रान्सफर व्हावं लागतं. लाईक दिस वे इन आवर इंटेस्टाईन. In our stomach or intestine require the presence of acidic pH. Our intestine require the presence of acidic pH. So from where that acidic pH is generated, in body itself the acidic pH is generated inside, and it is that acidic environment is transferred from one organ to other. आपण त्याच्यासाठी काही ऍसिड रोज पितो बाहेरून अशी गोष्ट नाहीये. Body itself generate acidic pH.